So what we'll do today is we're going to talk about what happens when DNA is damaged. Yesterday we described in some detail how the cell synthesizes DNA, and I introduced the idea that sometimes during replication of DNA there are mistakes that are made and they have to be fixed, and we'll see how they're fixed today. In addition, there are a whole variety of chemical insults that can occur in cells that damage DNA. Radiation of a variety of sorts will damage DNA. Oxygen, we live in an oxidative world, right? We are oxidative organisms. We have to have oxygen as the ultimate electron acceptor for our metabolism. And that's the good news. The bad news is oxygen has lots of other chemical effects in cells on a variety of macromolecules, including DNA. And we'll see how those the damage induced by oxidation can be fixed. We are sort of awash in a sea of various chemicals, some of which we made, some of which occur naturally, and some of which damage the DNA. And the term that is often used is called mutagenesis, meaning it generates or produces a mutation of some sort. And then, of course, as physicians, you're going to use drugs. So there will be iatrogenic damage to DNA because you're giving your patient drugs sometimes that cause some significant damage, especially in the case of the alkylating agents. Don't know what that means yet, but these are chemicals that are used to purposefully damage DNA and the DNA repair enzymes and the DNA binding proteins that we've been talking about all along. Okay? And in the case of cancer chemotherapy, the game there is the physician gives the drug to damage the DNA to stop the cancer cells. The cells respond by upregulating the DNA damage repair mechanisms. All the stuff we're going to talk about today in cancer cells can be tremendously upregulated or cells selected for that have very high repair capacity. So what happens? The cells become resistant to the cancers, right? And we talked about yesterday a case study of the patient PL who had the acute lymphoblastic leukemia, was given chemotherapy and became resistant. Why was that? Well, in part, probably because cells were selected for that had increased DNA repair. And so today our goal is to talk about how that can happen. So in order to do that, we need to talk a little bit about some chemistry of DNA. Now, what I've done is I've really narrowed this field down to a very few selected topics because the types of chemical modifications to DNA in cells is enormous. All right? So I've selected a few that are the most common or clinically the most important, and they're listed for you here. And we're going to go through those one by one and tell you about the types of mechanisms that will fix those processes. And here's the list of them. Okay, so our goal is going to be to know what those words mean, to recognize if I tell you a scenario, a patient scenario, which one of these processes that I'm referring to, or I may give you more clues about the process and you tell me which component, which enzyme, or which chemical reaction I'm asking you about. Okay, so here we go. So to remind you, here's the replication fork. Now this model, again, is based on E. coli, but what I say today applies to both prokaryotes and eukaryotes. The names of the proteins and the details of the repair mechanisms are a little different, but there's a lot of similarity, and I'll kind of point out where there's important differences. But starting with replication, the idea that I put forth yesterday, an important idea was that the DNA polymerase, here the green olive, in this case the bacterial enzyme is called POL3. The relevant enzymes in humans would be the DNA epsilon and DNA delta, or polymerase delta and epsilon. But in all of those cases, these polymerases do several things. They make the polymer, right? And that has some error rate. It's pretty low, 1 in 100,000. But if you're copying 6 billion bases in the human genome, that's going to be quite a few errors, right? but almost none of those errors get through. Why is that? Well, a very important point is that the DNA polymerases have the exonucleosidic proofreading, where they sort of sense the mistake, stop the reaction, cut the base out, put the proper base in, and then go on. Now that has a little bit of a finite error rate as well. Now the third step here is the new topic for today, which is strand-directed mismatch repair. So if something gets past these first two processes, it is frequently detected by strand mismatch repair. And so the product of all of these probabilities is here. It's about 10 to the 9. So that means that only 1 in a billion mistakes will get through, at least in the replication of the human genome. So if there's 6 billion bases, only 1 in a billion makes a mistake. You're going to get a couple of mistakes every time cells divide. Okay? So how does the strand mismatch repair work? Well, as you might guess from the name, this is a set of proteins. It's a complex set of proteins, most of which are not shown here, that detects a mismatch in the two strands, in the template strand, shown here in orange, and in the newly synthesized strand, represented here in red. And so what you see here is a little bit of a bulge. And this is meant to tell you, or to indicate that for some reason maybe the wrong base was put in there, and if it doesn't base pair, if you have an A trying to base pair with a G, okay, or a C, you're going to get a bulge. The DNA is not going to be the right shape. And there are proteins that basically ride along with the replosome. Okay, remember the replosome yesterday? We showed that trombone model, and the thing chugs along reading the DNA. Well, the repair enzymes are part of this complex. At least these repair enzymes are. And so they're going along essentially scanning the shape of the newly synthesized DNA. And in this example, this mut-S and mut-L proteins, these are the names of proteins in bacteria and yeast, okay, that do this job. There are homologs in mammals, okay, called MSH and MLH1. Basically do the same thing, but they have different gene names. Okay? So what do? Well, the mut-S, its job is to detect this mismatch, this misshape in the DNA. And the mut-L, its job is to then find a nick. Okay? So why is there a nick in the new strand? Where would this occur? What's the name of the structure in the lagging strand where there's nicks? Okazaki fragments, exactly. So the idea is that this structure can find these nicks, and then it knows, for one thing, probably this is the new strand, and because the way it's positioned on the replosome, we think that's how it recognizes which strand is which. Okay? Yeah?
So that's on lighting strand.
Great question. So how does it recognize the leading strand? Because there ain't no nicks. And the short answer is we don't know. The theory is, see, wasn't that easy? The theory is because of the way this thing rides along with the replosome, it's oriented so that the right strand gets used as a template. But that's actually not. It's a good story, but we don't know exactly. But it does fix the right strand, which is cool. But what happens then is the bad part is cut out, and then DNA synthesis reoccurs. Again, very much like it does normally, right? Polymerase goes along, polymerizes new DNA. So this represents kind of a structural view of how this process is thought to occur with the mut-S. So this is a dimer, meaning there's two molecules here. The green, you can see the green ribbons is one protein, and the other part of the dimer is the blue. And then the red, of course, is the sugar phosphate backbone, the white of the base is the DNA, and this is meant to represent there's a little bit of a kink here, and so this protein rides along with and kind of scans the DNA till it finds this error, and then the system fixes it. Now, it's a whole lot more complex than this. It's not just these two proteins. Okay, there's a whole bunch of things riding along with the replosome, but these are the two to remember, and they're mammalian homologs, MSH and MLH1. And the reason I want you to remember the mammalian homologs is that there's a very clear clinical correlate to this strand mismatch repair. There's a disease referred to as Lynch syndrome, or by its much easier to say name, hereditary non-polyposis colorectal cancer, HNPCC. So what's going on in this slide, and what does this have to do with strand mismatch repair? All right, so on the left side is actually a much more common type of cancer, familial adenomatous polyposis. Okay, this is kind of the most frequent type of colon cancer. And so what you see here is a chromosome smash where they've isolated the chromosomes from cancer cells and then taken pictures of them after they've performed what's called fluorescence in situ hybridization, FISH. What FISH does is you make a DNA probe, a piece of DNA that's fluorescently tagged, and you hybridize it, fluorescence hybridization, in situ hybridization. You hybridize it to the chromosome. And if it recognizes DNA sequence in the chromosome, it sticks. So each sequence is specific for genes in that particular chromosome. That's how you can identify which chromosome is which, or sometimes you can identify where the gene is in the chromosome. So that's an important technique in genetic testing. But anyway, the point here is they can see which chromosomes are which. All right? And as you can see, based on what we know about human chromosomes, is there's way too many chromosomes there. Okay? And so how did that happen? Well, it happened during the process of carcinogenesis. You have non-disjunction events. You have errors in DNA replication and repair. You got all kinds of problems going on in this type of colon cancer. So this is very diagnostic and characteristic of this type of cancer. Now, to contrast, the Lynch syndrome cancer has pretty much the right number of chromosomes, and they look pretty good, right? You don't see any big, long fusion, Robertson-type translocations. You just see sort of normal-looking chromosomes. So what's the problem? Well, the problem happens to be that these patients with this type of cancer, HNPCC, have mutations in the mismatch repair enzymes. And so they fail to repair properly, and that causes the cancer. Okay? So fundamentally different types of colon cancer. Now that's a very famous example that shows up on boards all the time. So you want to make sure you remember that. Lynch syndrome, strand mismatch repair mutations in MSH and MLH1. Now, that's the replication story. What about chemical damage? And as I pointed out at the beginning, lots of things can cause chemical damage, and here's a list. You don't have to memorize this list. A lot of it just is sort of common sense. Any chemical that's highly reactive is going to react with macromolecules in your cells. Your cells have a lot of DNA, so it's likely that the DNA is going to react. Now, but how to fix such damage, and what sort of damage are we talking about? So here we have to kind of go drill down into some specific chemistry that I'm going to want you to remember. So what you see here is just a little oligonucleotide, right? There's 4 nucleotides in this little stretch of DNA, right? Purine, guanine, right? Pyrimidine, cytosine, thymidine, adenine. See that? And so what the arrows are indicating are places on these molecules that are most frequently chemically modified. And the colors indicate what kind of modifications. For example, oxidation here in pink, this tells you that this bond is susceptible to oxidation. This methyl is susceptible to oxidation, this double bond, and so forth. the heavier arrow tells you that this bond is the most likely to be oxidized of these various types of structures. What else can happen? Methylation can occur at this nitrogen, okay, and this one, and this one. Hydrolysis can occur where this amine group can leave or this bond can be broken. And these two hydrolysis reactions lead to two particular types of modifications that we want to know, that we want to remember. They are depurination and deamination, where a C is converted to U, cytosine to uracil. All right, got that? So if I tell you a little vignette story on an exam, or if you see one of the boards, it will talk about hydrolysis of the amine at such and such a position in DNA, and that should trigger, oh, this is a DNA damage, it needs to be repaired. So let's talk about these two specific examples, deamination, depurination. So here you see this brown part. This is meant to represent the sugar phosphate backbone, so you can imagine it continuing along the screen, okay? And this guanine residue is a residue that's particularly susceptible to depurination. It's a purine, so depurination means the purine actually gets removed. And so this is a spontaneous hydrolysis. So hydrolysis reaction, you put water in, you change the reactants. This is spontaneous, non-enzymatic. This goes on all the time, okay? Fortunately, not at a real high rate, but at a significant rate. And the result then is you have a spot in the DNA where the purine is gone. Now there's a hydroxyl here instead of this glycoside bond. See that? So what does that mean? Well, what it means is you've changed the sequence of the DNA. You've induced a mutation. Well, you haven't. The water has. Okay? So a couple things can happen. During DNA replication, one strand, which is still perfectly fine, gets replicated, and you have a new duplex, and everybody's happy. But what about the other strand? When the opposite strand is used as a template, bad things can happen, because now the purine is gone, the DNA polymerase is chugging along, it's going to read a T and a C and nothing and then a T. So now you have a mutation where you have a point mutation where you've created a deletion mutation. A lot of times that might be fine. If that gets through, you know, we argued yesterday that 45% of the genome, or 90% of the genome, is in fact non-coding. It doesn't code for proteins. You think, big deal, I'll take those odds, you know. 90% of the time, this isn't going to matter. Or it'll matter in ways that that are not obvious. But this actually happens frequently enough that it does matter, and it must be repaired. And if it's not repaired, that creates significant numbers of mutations. Okay? So the other type of mutation that I want you to remember is this deamination. And the story here now, here's the DNA backbone. Here's the pyrimidine, a cytosine in this case. And you know it's a cytosine because of this amine group here. That undergoes spontaneous hydrolysis. And now cytosine becomes uracil. And you're thinking, okay, the four bases in DNA are A, T, C, and G. There's no U. There's no U in DNA. There's U's in RNA, right? But not DNA. So this is the wrong base. So what happens? Okay, so back to DNA synthesis. During replication, what happens now is you've got a base mismatch, and you get this sort of bulge or this misshape because U's and G's can't base pair properly, right? U will base pair with A, 2 hydrogen bonds, G with C, 3 hydrogen bonds. So the normal strand gets replicated, everybody's happy. The other strand gets replicated, and now look what's happened. Instead of a G in the new strand, it's become an A. Okay? So this purine G has been replaced with an A. So you've created a mutation that is referred to as a transition. That's the type of mutation, a transition. Okay, so we've created these problems, depurination and deamination. How do we fix them? And the answer is a process called base excision repair. And you can remember this pretty simply because you're going to fix one base, right? A base, base excision repair. And the way it's fixed is it's going to be cut out. That's what excision means. So what cuts it out? The bad base is going to be cut out by a family of enzymes called glycosylases. In this example, where we're going to fix the uracil, okay, replace it and turn it back into the cytosine that it used to be, this is a uracil glycosylation. But understand there's a whole family of these things, and all those sort of various oxidations and deaminations and various chemical changes I showed in the earlier slide, there's a whole family of enzymes that fix those particular changes. This is just one example that represents basically what happens. Okay, is that clear? All right. So what happens then is the glycosylase cuts out the incorrect U, basically cuts it off, and now here comes a new enzyme that's important and probably worth remembering. The AP endonuclease. What's AP? What does that mean? That refers to the term apurinic or apyrimidinic. In other words, it's a spot where either the purine or the pyrimidine has been cut out by a glycosylase. So it's missing a tooth here. So the AP endonuclease and phosphodiesterase. Why a phosphodiesterase? Well, what's the sugar? What's the backbone? It's a sugar phosphate backbone, right? Phosphoesters. So the phosphodiesterase cuts the sugar backbone. The endonuclease, and it's endo because it's working in the middle of the DNA, as opposed to exonuclease on the end of the DNA. The endonuclease cuts out this piece, and then it's repaired by the various DNA polymerase repair enzymes and DNA ligase. Put it all back together, and we're happy. Okay? So this just illustrates one particular base excision repair reaction, but understand that this applies, in principle, to many different chemical modifications that I summarized in the previous slide. Okay? All right. Now, that's to repair a single base, and it occurs after these chemical changes. It also occurs when any number of single-stranded lesions occur in DNA. Single-stranded lesions induced by a variety of chemicals and drugs and radiation occur very frequently. It's the single most common type of lesion. And the base excision repair pathway and components fix a whole bunch of those different kind of single-strand lesions, not just this type, but a whole lot of them. Sometimes the damage to the DNA is much more extensive, okay? And that's your clue in the question is if you see a question about extensive damage over long sections of DNA or large sections of DNA, many different modifications, bulky changes, that requires a different set of enzymes that you've already studied up on for today's TBL, and that is the nucleotide excision repair pathway. Now, the way I remember this is you think about oligonucleotides. That's what this thing is going to modify. In other words, a stretch of nucleotides, as opposed to base excision, which is one nucleotide. And what's shown here in the slide is one example of the way these nucleotides can be modified throughout the DNA molecule, and this is radiation. In this case, UVB radiation from the sun has a particular effect on pyrimidines. So when C's and T's are next to each other, UVB radiation can create cross-linking. When T's and T's are next to each other, it can also create cross-linking. Now, appreciate that given a high enough intensity, a variety of wavelengths of radiation will induce a variety of chemical changes. For today's discussion, we're going to focus really just on UVB-induced changes. Also understand that a number of drugs, particularly alkylating agents used in cancer therapy, doxorubicin that we talked about already, you've met in cancer therapy, these types of agents can create large sections of DNA damage because they chemically modify the DNA structure, or they modify the proteins that are binding to DNA. Those types of large damaged areas are also repaired by this same pathway. Okay? So don't get the impression it's only fixing UVB, but it's really generally useful for lots of different large-scale damages. All right. So again, you've studied this, so we're not going to go into this in huge detail right now. But the basic outline is damage is recognized by protein complexes that are stuck to the DNA, that are scanning the structure of the DNA routinely. In your reading, you learned that there's a process that goes along during transcription, right, when genes are actively transcribed. There's also sets of proteins that kind of scan the entire genome, even if they're not actively being transcribed, and they use slightly different sets of proteins. But the upside is they recognize something's wrong, probably based on structure. Nucleases then will cut out the piece of damaged DNA. Now, in this example, we only see one of these cross-linked pyrimidines. But again, understand that there's always multiple. There's a whole bunch, not just one. Helicase, right? In order to get the DNA strands apart, you got to have helicase activity. Comes along, pulls out the strand. And then now that you've got this big gap, how do you fix it? Well, DNA polymerase. It comes back, and then eventually DNA ligase. Put everything, put Humpty Dumpty back together again. Okay? So, in the TBL, we're going to go into this in quite a bit more detail with respect to the one genetic problem of xeroderma pigmentosa. Okay. Now, the other day I was telling you guys about homologous recombination, and I described this step in there where a double-stranded break is created. And during meiosis, the cells do this on purpose, right? There's an assembly of protein structures that purposefully cut the DNA and recombine the chromosomes to create genetic diversity. So that's a good thing. But double-stranded DNA breaks also occur, again, ionizing radiation, alkylating agent drugs, mutagenic chemicals in the environment. They occur for a lot of reasons. And of all the types of DNA damage, this is potentially the most dangerous. Okay? Think about our ongoing story about patient PL who has the Philadelphia chromosome, who had that big translocation. Okay? What was the first step, do you reckon, on the path to making that translocation? You have to cut the DNA, right? Double-stranded DNA cut. Most translocations are thought to start with a double-stranded DNA cut. So how do you fix such things? There's two basic ways. One way is fast and easy, but error-prone. And that involves chewing back the sticky ends. So in this example, you can see where the ends are not even. So there's some overhang. All right? In molecular biology, we call those sticky ends because they'll tend to rehybridize together. So what happens in a cell is these ends get chewed off by nucleases, exonucleases, to form blunt ends. Okay? Same length. The blunt ends can be very efficiently rejoined, okay, by what a process called non-homologous end joining. So non-homologous means that it doesn't depend on sequence homology. Okay, it'll happen pretty much anywhere. All right? Now, the good news is, again, because so much of the genome is not protein coding, you can get a double-stranded break, snip off a few nucleotides, paste it back together, and everybody's happy. Right? But sometimes that occurs in a protein coding region, and then you create a deletion mutation. So that's the bad part of it. Now, turns out that there's also kind of an interesting positive aspect to this that's used by the immune system. You realize that when you get an infection, you raise antibodies, right, against the infection. And there's about 50 trillion billion things that you can raise antibodies against. So how does the cell, how do immune cells figure out, or how do they generate so many different kinds of antibodies? And part of the answer is non-homologous end joining. And there's a process you'll learn about in immunology where the variable region of the antibody protein is variable because the genetic sequence is variable, in part because of this non-homologous end joining. So that's a good aspect of this process. Okay? All right, so this is fast and dirty, but inaccurate. You're losing bases, you create problems. A more accurate way to repair this double-stranded break is the process of homologous recombination. Now, we know exactly what this is. We talked about this in detail yesterday. But again, the setting, the timing is profoundly different. During meiosis, homologous, double-strand breaks and homologous recombination occur on purpose to generate diversity. In the case of repair, it's a little trickier because most of the repair occurs not in germline cells, but rather in mitotic cells and somatic cells that are just going along and doing their thing. So to get homologous recombination, you think, well, wait a minute, we've got to get the two chromosomes together, okay? Because you have to have the broken DNA, and you got to have the intact DNA in order to repair the broken DNA. So you've got to get the two chromosomes together. So when does that actually happen in a cell? Well, not very often during interphase, but it does happen during mitosis. And so that offers an opportunity where you could get homologous recombination during mitosis to fix the damage. And that's what we think happens. So just to remind you, again, what happens during recombination, if there's a break, again, in this point in the discussion, a break caused by some damage, then exactly the same thing happens as it does during meiosis. There's nuclease chewing back to make sticky ends. The sticky end goes and finds its homologous region in the other chromosome by invading and creating what's called this D loop or a DNA synapse. And again, remember, this is assisted by recombination proteins, REC-A in bacteria, RAD51 in the BRCAs, and other proteins in mammalian cells. A little bit of DNA synthesis occurs, right, to fix the piece that you chewed back. And that happens on both strands. This forms the holiday junction, which then can be resolved. And if it's resolved, I'm going to skip ahead to remind you there's two ways the holiday junction can be resolved. During meiosis, where you're getting recombination on purpose, you resolve it like this, right? Green, red, red, green. During DNA repair, you don't want that. You don't want to recombine. What you want is you want to go back to the same chromosomes. So the holiday junction is cut in a different direction to give you back the chromosomes you started with, thereby repairing the double-stranded break. Does that make sense? Same basic biochemistry, different timing, different purpose, different stimulus, different reason for doing the repair. Okay. Now this, unlike the non-homologous end joining, this process is highly specific. Okay? It's low probability, but it's highly specific and very accurate. You make a mess, this will fix it. All right. So, we're going to wrap up here by summarizing what do we need to know about all this stuff and how does this relate to medicine and pathology. So I've prepared for you a table. These are kind of classic examples of inherited syndromes or diseases that involve defects in the DNA repair. Excuse me. So you can imagine defects in DNA repair have profound implications. One we've already talked about. In colon cancer, particularly the Lynch syndrome, HNPCC, when the mismatch repair system isn't working right, errors will creep through, replication errors will creep through, if there are mutations in these gene components. Okay? So they're highlighted in red, which means I'm going to put these on the test and they'll be on the boards almost for sure. XP, we're going to talk about today, that's going to be on the test. And on other tests, you can, in my test, you can expect maybe a question about some of these other syndromes. Now, you don't have to memorize in great detail the underlying biochemistry of what's happening here, but you should at least recognize if there's a story about breast cancer and homologous recombination, and I ask you what's the likely culprit, you should come up with BRCA2. See what I mean? Okay. And you'll see if you look in board review books that almost all of these characters will show up at the end of the molecular biology section as syndromes that you should recognize and appreciate have something to do with DNA repair and maybe be able to identify exactly what step in the DNA repair. Clear? Good? Okay. Okay, so this, we're going to wrap up early on this section, take about a 10, 15-minute break, and just to preview what happens later in the day, we'll go over and have our first team-based learning exercise focusing on this xeroderma pigmentosum. Now, it was pretty technical, okay, lots of detailed stuff in there for you to learn, and you might say, well, how prevalent is this disease? And the answer is not very. It's a very, very rare disease. I would guess that in your training here at South, you may never see anybody with xeroderma pigmentosum. But it's a very famous example that illustrates a couple of important things. It shows you how nucleotide excision repair works, how it happens in some detail, and it really illustrates that it's really, really important. Because if you look at that phenotype in these afflicted individuals, it's pretty dramatic. It's pretty debilitating. Okay, so it has some, medical drama to it. But it has some sort of humanism aspects too that we don't normally talk about in biochemistry. Is you understand these children are very, very restricted in what they can do. Right? They have to have these extensive covers, and their families have really complicated care plans to take care of them. Okay, so this has implications just beyond, which XP complement type we're talking about. It has implications for therapy and for the family dynamic as well. And so that's kind of why I decided to do this sort of very highly focused and fairly technical case example. So let me ask at this point, are you guys okay for the TBL? You got questions about what's going to happen? Got a clear view? We're going to do the IRAT. I kind of gave you some clues in the reading to know this. That's going to be on the IRAT. Just as a clue. Okay, that makes sense? Everybody clear on that? There were a couple questions yesterday about the how do you define the complementation group? Is that clear what a complementation group is? From classic genetics, complementation assays are a way before we could do high throughput sequencing and just drill down to the DNA sequence to figure out what gene is responsible for which phenotype. The classic approach is complementation. So you, in effect, you add back the gene, a candidate gene that you think is responsible for the phenotype. If that gene's missing, add it back. If you get the phenotype, then voila, that gene was necessary. That's sort of what it boils down to. Okay? Now, truth be told, modern era, a lot of that stuff is actually being replaced by PCR and RFLP and high throughput sequencing to just drill down and say, okay, get some DNA and figure out what's wrong with this person. That's kind of the new way to do it. But this example, I think, is a nice illustration of the classics. And again, the tests are usually 4, 5, 6, 10 years behind where the leading edge of the biomedical science is. So it's still worth doing this. All right. Anything? You okay with the TBL? You good to go? All right. Well, let's take about 10 minutes, and then we'll come back and talk about RNA.